Amanda for that nice introduction. I'm really excited to be here today. So thank you so much for inviting me. And I'm a bit sad that I'm not there in person because I'm desperate to come and have a look around and chat and hang out with you guys, but another time maybe. But anyway, so I'm really excited to be here and to be talking to you all and sharing some of the work that we've been doing over the last few years, looking at soil fungi that form these partnerships with plants, looking at their significance in helping plants colonize the earth over 500 million years ago, and how they've responded to and helped to shape Earth's changing climate ever since. So we're only going to be spanning 500 million years of Earth history here, so I'll try not to fill up the whole hour, you know. Anyway, okay. Okay, cool. So our planet is often referred to as a blue planet, thanks to the extensive coverage by the seas and oceans. However, I think instead we should be referring to it as a green planet, okay, because this reflects better the unique, diverse, and biologically critical plant coverage on Earth. In fact, actually 31% of Earth's surface is covered by forests on their own. And of the 550 gigatons of carbon that's on Earth, which is that critical element for all life, plants are responsible for 450 gigatons of it. Now that's really hard to visualize. So just for a bit of contextualization, that mass of carbon, 450 gigatons, is the same mass as a billion African elephants, which is obviously much easier to imagine, right? Anyway, it wasn't always like this. Earth wasn't always green. If we travel back in time about 500 million years or so, plants hadn't yet made their move onto Earth's terrestrial surfaces. And the only sort of terrestrial photosynthesis really happening on Earth was kind of happening at the water's edge. Potentially something a bit like this, where you can see you've got algae, cyanobacterial mats, giving some color to those marginal zones at the edges of rivers, lakes, seas, etc. So once plants made that transition from an aquatic to terrestrial existence, however, things started getting really exciting. So this is an artist's representation of the landscape of the Scottish village of Rhynie around 410 million years ago. And you can see the colors and emerging diversity of plant and fungal life. So we have these huge fungal structures in the background here. These are actually prototaxites, which we now think is a fungus. And you can see it extending way up higher than any of the plants present in that landscape. But how do we know it looked a bit like that? Okay, well, this is Rhynie today, and it looks on the face of it pretty unremarkable. However, if you scratch the surface of those surrounding fields, you uncover some incredible fossils that depict the plant, fungal, and animal life present around 410 to 412 million years ago. And those fossils are there largely thanks to the unique environmental conditions in Rhynie at that time. So this is an example of a Rhynie chert fossil plant, and it's pretty incredible. You can make out bifurcating stems and what we think is sporangia born at the end of those. And, you know, they look like a bit like plants. But if you look even more closely, this is an Agolophyton major. So this is a slice of the axis of Agolophyton major. So these axes sort of snaked along the ground. And if you slice them up and look more closely, you can see how incredible the preserving power of Rhynie was. You can make out like individual cells within that section. Even more amazingly, if you look more closely at this smudgy, browny, grey sort of area and zoom in on your microscope, you can make out little structures inside those cells that look like this. And these look incredibly similar to the arbuscular mycorrhizal fungal structures that most land plants form in their roots today. So this is a section through wheat, and you can see the fungal structures, and you can see how similar they look to those ancient 400 million year old fungal fossils here. So I think everybody on this call, I don't know how familiar you are with mycorrhizas, so I'm just going to do like a little bit of an overview of what they are and why they're important to plants. But mycorrhizas are what we call the partnerships formed between soil fungi and plant roots. In this case, I'm focusing on the arbuscular mycorrhizas, all right, which is a group of fungus that forms symbioses with roots of more than 70% of land plants today. Once inside the roots, so they germinate in the soil from these spores, they detect signals from the roots, and then they grow through the soil and they hit the plant roots. And once they hit those plant roots and grow inside, they form these distinctive structures that we just looked at in the previous slide. And it's through these structures, the plant is able to assimilate mineral nutrients that the fungus has scavenged from the soil. And in return, the plant actually transfers organic carbon in the form of sugars, fatty acids, and lipids that it's converted from what it's fixed through photosynthesis. So you have this bidirectional exchange of resources between plant and fungus that's characteristic of our vascular mycorrhizal partnerships. Now, although we usually think of these symbioses as being true mutualisms, where both parties benefit from the partnership, you can see here, this is represented in this cartoon here with both looking pretty friendly and shaking hands. They're actually really flexible in terms of their function. Whoops, oh my goodness, don't touch that. Ranging from these true mutualisms that I just described all the way through to full parasitism, okay, by either the fungus or the plant. So this plant, for example, is known as a mycoheterotroph, all right? And that means basically it gets all of its nutrition from its fungal partners, even going so far as to lose its chlorophyll and therefore the means of synthesizing its own organic carbon products. Various factors influence where on this continuum between mutualism and parasitism a specific symbiosis might lie. And these include factors are abiotic, so things like soil nutrient availability, soil type, disturbance, CO2 concentration, as well as biotic factors such as competition, genetic diversity, and compatibility of the partners themselves. So we have these flexible symbioses that exist on a continuum ranging from mutualism all the way through to parasitism. So if we go back to our early plant AM symbioses, it seems logical on the face of it that by associating with mutualistic symbiotic fungi, such as our vascular mycorrhizas, those first plants to get onto the land could well have gained an advantage, bearing in mind that these plants, you can see in this beautifully drawn picture, these plants have got no roots, and there's very little plant available nutrients in that primordial soil crust present at that time. So you also don't forget, you don't really have soil as we know it yet. But considering AM symbiosis, so when I say AM, I mean our vascular mycorrhizal, sorry, I just slipped into acronym zone there. But considering mycorrhizal symbioses really span this mutualism, parasitism continuum in terms of their function, and as we've seen time and again when studying modern mycorrhizas, that presence of those structures in plant roots actually gives away very little in terms of clues in what they're actually doing. Did those early plants with these fossil structures in their root, or what are now fossils, did those early plants actually gain from their fungal partners? Or were these fungi merely passengers just hitching a ride onto land with those early plants? So in order to try and piece together this puzzle, and because we don't have time machines, we need to look at modern relatives of the earliest land plants. And so in this case, we look to the bryophytes as being similar to early plants in terms of lacking vasculature and roots, and as being the earliest divergent branch on the land plant tree of life. So using liverworts in association with our vascular mycorrhizal fungi, recent research has shown that by associating with the fungus, the liverworts actually benefit in terms of photosynthesis. So this experiment here is using mycorrhizal liverworts, that's the white bars here, are non-mycorrhizal liverworts at 2 different CO2 concentrations. And you can see under both CO2 treatments, by associating with mycorrhizal fungi, they benefit in terms of photosynthesis, in terms of growth. So the white symbols generally grew bigger than the, oh, hang on. Anyway, they benefit in terms of growth and then also in terms of fitness. So the black bars, sorry, they switched colors halfway through, that helps. So the black symbols are the mycorrhizal ones in these 2 figures. So they benefit in terms of photosynthetic rate, in terms of biomass, and in terms of fitness. So this is the number of gemmae that are produced per liverwort in the experiment. And the gemmae are just the little reproductive structures that are born in cups on those plants. Okay, this more recent work, very slightly more recent at least, this started to unpick actually why and how those plants are benefiting. Why were those liverworts getting these growth and photosynthetic benefits from the fungal partners? And if you look at the liverworts Pricea and Marcantia, which is these two sets here, we're looking at the black bars, which is an elevated CO2 concentration, and the white bars are an ambient CO2 concentration. You can see you get a benefit in terms of phosphorus in the plant shoot in return for carbon invested in the fungus. So together, these experiments represent the first evidence that these fungi actually do benefit the liverworts they're associated with, and that it's they're benefiting them in terms of carbon for nutrient exchange between these different symbiotic partners. Okay, so this kind of is important, well, it is important because it suggests that those mycorrhizas observed in those fossils of ancient plants are likely to also have been beneficial. And thus the idea that mycorrhizal fungi played a critical role in colonization of the terrestrial environment by plants becomes much stronger. However, and there's always a but, right? In 2011, Martin Birartondo and his collaborators made this really exciting discovery. So they were examining the fungal partners of this early diverging group of liverworts. And what they discovered was that these liverworts here were not actually associating with our buscular mycorrhizal fungi as they expected, but instead they were forming symbioses with a totally different group of fungi belonging to the fungal subphylum known as the mycorrhizal. So given the placement of these liverworts in particular on the land plant phylogeny, being right at the very bottom, coupled with the placement and capabilities of mycorrhizal fungi, this discovery actually gave rise to a whole new hypothesis for the greening of the earth, that actually it was these mycorrhizal fungi that may have played an important role in helping plants get onto the land. And potentially these fungi were the ones that represented the ancestral mycorrhizal type. So this hypothesis actually relies on mycorrhizal associations being mutualistic in at least the same way as the arbuscular mycorrhizas, but it was completely untested. We had no idea these fungi were even there, let alone we had no idea what they did. So we decided to test it, and in order to do that, my collaborators and myself, we traveled to the South Island of New Zealand, and here you can see Jeff Duckett, Martin Birartondo here, myself, Will Rimington, Sylvia Pressel and Grace Hoysted, hard at work in the field. We went there to find the liverworts that associate only with these fungi so that we could experiment with them and find out what that fungus was doing. So I'm introducing the lovely liverworts here, Troibia lacunosa and Haplomytrium gibbsii. So these are members of the Haplomytriopsida, which is a very basal clade of liverworts in the land plant phylogeny, and they only associate with mycorrhizal fungi. So as well as those two liverworts, we also collected another two types of liverwort that also associate with mycorrhizal, but these guys actually associate with mycorrhizal and arbuscular mycorrhizas at the same time. Okay, so this is Allosonia, and this is Neohogsonia, and I'm sorry they're not the best pictures, but they weren't terribly good models, to be honest. Anyway, we brought these fungi back to the lab in the UK and set up microcosms to track the movement of carbon and nutrients between the partners. And the microcosms that looked like this in diagrammatic form, and in real life, they looked like this. So these microcosms are based on ingrowth core system that was pioneered by Dave Johnson at the University of Sheffield. And they consist of these windowed plastic cores with this mesh that covers those windows. And that actually allows fungal hyphae to grow inside the core, but it means the rhizoids of the liverworts aren't able to because the mesh is too fine. So by doing this, you create this compartment within your pot that only fungal hyphae can get into. And so you can then introduce different nutrients, isotope tracers, and so on, and actually track the movement of those nutrients and carbon between the two partners in the microcosm. By pairing your cores within your microcosm and rotating one of them, you can exclude the contributions made by microbes and passive diffusion of substances through the soil solution, because you're actually severing the fungal hyphae. You're severing the fungal hyphal connections, so you can subtract one from the other, and in that way you can find out what the contribution of the fungus is to that system. So using these microcosms, and we used them at 2 CO2 concentrations. So the first was replicating a modern day climate, so 440 parts per million, which seems a little high, but if you get close to the ground, that's more similar to what it would be down there. And second one was 1500 parts per million, and this was supposed to be simulating a Phanerozoic style CO2 concentration to really test how far could we push this symbiosis and how does it affect that placement along that symbiotic continuum. So we grew these and we released a 14 CO2 pulse into that microcosm, and then using these fungal ingrowth cores, we trace the movement of carbon from the plant to the fungus within that core. So this graph is horrible, and I'm sorry, I can't think of a better way of showing it, so I'll just break it down. So here we're comparing the amount of carbon that is going from the plants to the fungus in 3 different types of symbiosis with liverworts. So we have our types of liverworts on the bottom down here, and here are pictures, just in case you want to put a face to the name. These ones associate with Mucormycotina. These guys, Glomeromycota, this is the arbuscular mycorrhizas. And then these guys at the end here, these are dual colonized plants. They have both types of fungal symbiont present within the thallus of both of these liverworts. And the first figure here, this shows us the amount of carbon that is allocated to the fungus within the core. And this is a percentage of the total amount of carbon that was fixed by the plant during the time it was being labeled. So it's very low in all cases, which is great for the plant. It's not having to give away very much of its photosynthate, basically. But what we see from this is, firstly, yes, Mucormycotina fungi gain carbon, albeit a very small percentage from their fungal partner, and that in terms of percentages is roughly applicable across CO2 treatments and across symbioses. Okay, however, if we look at it in terms of absolute amounts, so this is then the absolute amount of carbon that's going from the plant to the fungus in each symbiosis, a slightly different picture emerges. So it seems the CO2 atmosphere doesn't make a huge amount of difference. If anything, it goes the way you might expect. A higher CO2, more photosynthates available, more carbon goes to the fungal partners. That's, oops, at the black bars. But what's really interesting is that the dual associations seem to be gaining. I've got to stop touching that. The dual associations are actually costing the plant a lot more carbon than either of the single colonizations that are pictured here. And I just want to point out that this is a log scale, so we're going up by an order of magnitude each time. So this is a really significant cost to a little liverwort in terms of carbon. So then we wanted to look at the exchange, the other side of the exchange. So we wanted to look at the assimilation of soil nutrients. So we used a 15N and a 33P labeled isotope tracer in those same microcosms at both of the CO2 concentrations again. And again, apologies for the horrible graphs, but first thing to take home, this top figure, again, we've got the 3 partnerships along the top, different liverworts on the bottom, and we've got the 33P tracer. So this is the amount of phosphorus gained from the fungus in the plant. And then the bottom one is nitrogen, and we don't have the data for the arbusculars in these systems. But the first thing to notice is that, yes, Mucormycotina fungi, that's these guys here, they transfer both phosphorus and nitrogen to their plant partner. So this is a mutualistic relationship between the plants and the fungi. We also found that the dual colonized plants also transferred phosphorus and nitrogen to their fungal partner. But what perhaps was really interesting, particularly I thought at least, was that these dual associated plants actually gained more nutrients than the single colonized plants under the ambient CO2 atmosphere. And this might explain why they've got these both types of mycorrhizal association, why they've persisted for so long. Okay, so just to summarize those results right now, we have these dual fungal associations costing much more in terms of plant fixed carbon than either of the single fungal associations at either of the CO2 concentrations that we tested. The liverworts that associate with the Mucormycotina fungi gain more phosphorus under modern day CO2 than those that associate only with arbuscular mycorrhizas. there seems to be no difference in fungus to plant nutrient transfer between symbioses at the high CO2. And the exchange efficiency appears to be greatest for liverwort mycorrhizae associations at the lower CO2, comparative across and it's comparative across symbioses at the high CO2. So these observations have driven this hypothesis that we're investigating now, which is why have these associations persisted for so long? Currently, it seems that they are more efficient at the more modern lower CO2 concentrations in non-vascular plants than either of those single colonizations, which kind of creates this physiological niche for that particular symbiosis. And it also seems that nitrogen plays a role in this that was perhaps not expected. So we wanted to test why, what is the role of mycorrhiza symbiosis in nitrogen assimilation particularly. And we wondered if it was something to do with their potential capabilities of saprotrophic nutrition. So mycorrhiza as a fungal group contains loads of saprotrophs and only this one grouping of symbionts. And so we thought, well, if it's in that group, it could well still have saprotrophic capabilities. So we wanted to test that. And so we provided using microcosms like this, which are just like one step up in complexity from the ones we looked at before, where we actually include, rather than injecting 15N ammonium chloride, we bound the 15N into this necromass of single-celled algae, so that's chlorella, and we either let the fungus have access to that algal necromass or not. And we basically measured the uptake of that organically bound 15N alongside inorganic 33P and a 14C trace to measure the flow of carbon into the fungus. So these are the figures, this is what we found from that experiment. And this is looking at phosphorus. So we have total phosphorus here, high CO2 again, and low CO2. And then this is concentration in plant tissues. This is normalized to plant biomass here. And basically we found no difference between the symbioses essentially in terms of phosphorus transfer at the lower CO2. And then at high CO2, the pattern of phosphorus uptake was pretty much the same as we saw previously, which is good, right? Because that means we repeated it and it still did the same thing. However, the availability of organic nutrients, so where they had access to the algae in the core, it seemed to promote phosphorus uptake in mycorrhiza associated plants. For nitrogen, though, this is really interesting. We found that both CO2, so again, you've got high CO2 in these two figures, low CO2 in these two figures, total nitrogen, it said, oh God, total nitrogen and nitrogen concentration on this side. So we found at both CO2 concentrations that more of the 15N was accessed and transferred to the plant when a mycorrhiza partner was involved than when it wasn't. Okay, and this suggests that there may well be some functional complementarity between the two symbioses in liverworts, and so potentially also in early plants. And this adds weight to the idea that this second group, this mycorrhiza fungal group, were also important to early plants, and that there were actually symbiotic options available for the colonization of land by plants. Okay. So what about vascular plants? We've kind of only looked at non-vascular plants, but what about, what some people would say are more important, but certainly much more diverse now, these vascular plants at this end of the plant phylogeny. Well, having done a bunch of sequencing across different plant groups, we've found that yes, you do get these associations in vascular plants. We've also got evidence now for flowering plants. But we have no idea what they do in vascular plants. And having looked at vascular mycorrhizas, we know the functioning in vascular plants versus non-vascular plants, it can be a little bit different. So we moved on to this early diverging vascular plant. It's a club moss. This is Lycopodiella inundata. Okay, and this work has been done largely by one of my postdocs. This is Grace Hoysted. And she's been working a lot with Lycopodiella inundata. Okay, and this is a lovely little club moss, and it forms associations with mycorrhiza, but not, to our knowledge, ever with arbuscular mycorrhizas. So we have this really nice experimental system ready for us to use. So if we look at the roots, we can see characteristic mycorrhiza structures, okay? And by sampling around, so which one's the, I think this is the Lycopodiella inundata root. So these are the characteristic structures. You've got these fan-like branches, these little lumps within and across the hyphae. But then when we sampled around neighboring plants, so things like the liverworts that were there, grasses, rushes, we also see those characteristic structures, suggesting that, you know, these are much more widespread than we previously thought. So sequencing of those fungi actually revealed that they were the same fungi and they were shared throughout the community. And this, really demonstrates and confirms to us that these mycorrhiza fungal associates are really widespread and not limited to those weirdo early diverging lineages of plants. But what do they do in the plants? What do they actually do? So Grace carried out a whole bunch of isotope tracing experiments, very similar to the ones that I did with the liverworts. And using the same isotope traces, she was able to show there's abundant carbon for nitrogen and some phosphorus exchangeable between partners. So you can see here, 2 CO2 concentrations. She's got lots of carbon being transferred from plant to fungus. And in return, the plant is gaining a bit of phosphorus, but a lot of nitrogen. Okay, and this was kind of unexpected. It was also fairly unexpected that this symbiosis appears to be completely unaffected by atmospheric CO2 concentration. So just to firm up those results, we also looked at the stable isotope signatures. So Gerhard Gebauer and his PhD student Philippe Giesman, they sampled the Lycopodiella inundata, that's the yellow triangle here, and also Juncus bulbosa, which we had found the mycorrhiza present in the roots of here. And they looked at the delta 13C and the delta 15N isotope, natural abundance isotope signatures. And what they found was that it was very distinct in terms of 15N. Okay, and this suggests that they were getting their 15N in a different way to these plants that weren't associated with mycorrhiza. Okay, so the accumulation and the enrichment of 15N in these plants suggest it was coming via a fungal partner. And if you're interested, there's much more detail on that in this paper. So we have this widespread nutritionally mutualistic symbiosis between plants and mycorrhiza across the entire land plant phylogeny. So building on our discovery of the symbiotic mycorrhiza in 2011, they were linked to the much neglected fine root endophytes in 2017 by another group in Australia. So fine root endophytes have always been assumed just to be another type of arbuscular mycorrhiza. And that's mostly because looking down a microscope, they look similar, okay? However, we now know they're a distinct fungal group and that their functional significance appears to be complementary, but distinct to that of the arbuscular mycorrhizas. So we've shown that these mycorrhiza fine root endophytes, that we're now calling them, are mutualistic. We've shown that they're CO2 responsive in non-vascular plants. And perhaps most importantly, they seem to be everywhere, okay? So yeah, we know that they're everywhere. And we know that in non-vascular plants, at least, they respond to changing atmospheric CO2. But what about feedbacks? What about the effects those fungi themselves might actually be having in the opposite direction? What effect might they be having on global climate and CO2 concentrations themselves? So it seems like a bit of a mad question to ask, right? But just to unpack how they may have had a role, because like I say, it does initially seem to be a bit of a mad idea that fungi could have influenced global climate. Now we know that plants gain soil nutrients from their fungal partners in exchange for carbon fixed through photosynthesis, okay? The fungi get some of these minerals by interacting directly with mineral particles in the soil. So this electron micrograph, you can see fungal hyphae traversing the soil particles. And as they do so, they trench along it, dissolving nutrients from that rock directly, and then they assimilate that and pass it back to their partner. So here, this is a mineral chip, and you can see an AM fungal spores have germinated, and it's traveled along the mineral chip, trenching its way through that as it goes, actually eating the rock itself. So more nutrients going to the plants through their fungal hyphae, means you can have more plant growth, all right, which means you get bigger plants, and those bigger plants have got a greater demand for CO2 for photosynthesis. And this results in a greater CO2 drawdown from the atmosphere. So by teaming up with Ben Mills and Sarah Batterman from the University of Leeds, we added this mycorrhizal phosphorus uptake box onto Ben's paleoclimate model. So it's based on the COPS model, if that means anyone to anyone here. But basically it's a box model simulating the processes and cycles that dictate Earth's climate. So we added this mycorrhizal P box onto that. And we parameterized it using the data that we'd gained on carbon for phosphorus exchange from experiments with liverworts. And this is what we found. So this figure here, this is terrestrial MPP relative to today. The black line is if mycorrhizas don't exist throughout time. So this is a geological timescale we're looking at, and this is millions of years before present. The blue line represents if we were only dealing with arbuscular mycorrhizas from the beginning of time. And the red line represents if we're only dealing with Mucormycotina fungal associates through time. And you can see these different types of plant fungal partnerships actually would have had a huge impact on NPP across time, with it either being much greater than we thought, if it's arbuscular mycorrhizas, or it might have been much lower than been predicted if it was all Mucormycotina. If you look at the oxygen concentrations, so a Mucormycotina only world, we could have delayed the rise of oxygen for a few 100 million years, or we could have accelerated it if we're talking an arbuscular world. And in turn, CO2, it mirrors that pattern, with a Mucormycotina dominated world being much higher CO2 concentration for much longer than if we had an arbuscular world. And this translates then into quite different average global surface temperatures, with Mucormycotina world being much hotter for much longer than a arbuscular mycorrhiza dominated world. And so, I mean, this is like a huge extrapolation from just some very small plants and some very small limited experiments with those tiny plants. But I think what we really wanted to just show with this, firstly, it was a lot of fun, but also the message is just, you know, fungi matter, and we need to consider these when we're thinking about climate and climate regulation. So this is just a bit of a sneak preview now of some of the work we've been doing in the last few months on this subject. So we recently were lucky enough to get a European Research Council grant awarded. And essentially through this project, MycoRev, we're asking these key questions, which we're hoping to address in the context of modern terrestrial ecosystems. So which plants do Mucormycotina fine endophytes actually associate with? Are they really as widespread as we think they might be? Where are they occurring? Why are they there? What is that fungus doing in those plants, and how is it doing it? And lastly, how is that moderated by the environment? So how does it respond to various changes in soil, atmosphere, and water? So these are some early results from some of the lab work we've been doing during lockdown. So you'll bear with me that they are a little bit preliminary. It's kind of amazing we've been able to do anything at all, but I thought I'd just share them with you because I'm quite excited by it. Basically, we've been able to isolate the fungus from root sections of Lycopodiella. So we've been able to get that fungus growing onto agar out of the roots, which has like opened up a world of experimental tractability for us. And we're able to establish these monozenic systems where we're growing plants, like real plants, like clover, proper higher stuff, with the fungus in culture, and we're able to then manipulate it and actually find out what the direct role of the fungus is in plant nutrition. So these microcosms are really important and open up a world of stuff to us because they don't have contamination by any bacteria, and there's no other fungus present. So we can really quantify the role of Mucormycotina in modern plant systems. So the root colonization we see in these experimental systems is very similar to what we see in wild plants. And you can see it is quite distinct to arbuscular mycorrhizal colonization. We're kind of limited to this quite crude trypan blue staining because obviously we can't get in to use our scanning electron microscopes and stuff. But we do see the characteristic structures. We see that nice hyphal fan-like branching. We have the fungal lumps that we've seen in the previous wild collected plants, and we have these fungal coils within cells, which is super nice. And then just before we got totally locked down, we were able to actually do a really cheeky, quick isotope tracing experiment using these systems. So we cut a trench to sever the connections between the fungus and the plant roots, and we introduced a 33P labeled well. We also introduced 15N label into that well, cut into the media, and then the trench in some of them will have severed the hyphae, and in other ones we let it keep growing, and then in other microcosms we had just plants growing on their own with no fungi, no trenching or anything. And what we found is that there was lots of phosphorus and nitrogen transfer from fungus to plants. So this confirmed the work that we'd done with wild collected weird plants in soil-based dirty systems, that actually the fungus is having a direct role in transferring nutrients. But I think most exciting thing for me looking at these data was actually the units, right? So phosphorus, we're getting a little bit, we're talking nanograms or less than half a nanogram in the best circumstance. But when we talk about nitrogen, we're getting like micrograms, like orders of magnitude, huge amounts of nitrogen being transferred from this little well to the plant tissue. However, we don't know about carbon yet. We've just done a labeling last week actually with carbon, so we should have answers about that soon. And also non-leguminous plants, right? We haven't done it in non-leguminous plants yet. So there's a whole world of discovery waiting for us with that. Which brings us up to sort of like modern day stuff. So we've come all the way from early Devonian all the way through to modern day. And today, more than 90% of land plants have fungal symbionts in their roots. And that includes crops, okay? And that's really interesting and important, but particularly in a sustainability context, because we know that in the wild, mycorrhizas, they can act to increase plant access to soil nutrients. They also have other non-nutritional roles, so things like enhancing plant defense against disease and pests by priming plant defense signaling systems. And they also can act to increase drought tolerance and resistance. But our challenge now is to translate all of that work that's been done in wild plants into crops and set it in the context of global climate change. So can we make the most of soil fungi to help feed the world? I'm sure everyone's very familiar with graphs like this, but what we have here is the yield of wheat increasing steadily through time from the 1980s all the way through to present day. However, what's really a bit disturbing is that actually the leveling off of this curve in more recent years, okay? And that's disturbing because when you compare it, this doesn't have the label on, but this is the amount of fertilizer added to fields every year, okay? And what we can see is from about the late 90s is that actually you don't have that increase in yield in line with the increasing fertilizer application, okay? And that is worrying on a number of fronts because application of fertilizers, pesticides, and so on has driven the green revolution and the dramatic increases in crop yields since the 1950s. What's also a bit disturbing is that actually the depletion of resources that we use to make particularly finite resources like phosphorus, increasing energy prices, all of these things against the background of increasing human populations, we're now facing this food security crisis. And so some of us are turning to mycorrhizal fungi and wondering, are these naturally occurring soil fungi that can help plants access nutrients that are already in the soil, are they really a sustainable solution for this crisis? We know that both, I'm going to use wheat and barley as like the main study systems here, because in the UK, that's a very relevant crop to be talking about. So we know that the main cereal crops in Europe are capable of forming mycorrhizal associations. However, what we also know is that this ability seems to be very variable. And that's largely a result of selective breeding for other traits that has like inadvertently selected against being mycorrhizal. So for example, selecting for responsiveness to fertilizers, disease resistance, that actually selects against forming good functional mycorrhizas. So we have this huge variability in the capabilities of forming functional mycorrhizas in modern wheat and barley. So What we don't know, though, is how this translates to function. So just because, again, I've said it previously, but just because fungal structures, you can see them, it doesn't necessarily mean they're functional. So the ones that are getting well colonized, we don't know is it actually doing anything good or not. So these are the key questions that we're asking in some of the other research that we've been doing recently. Is firstly, what are these symbioses doing in crops? We know in wild plants, so Osmunda is a fern, Plantaga is just a forb. We know in wild plants, mycorrhizas do carbon for nutrient exchange, and actually this can be very heavily influenced by atmospheric CO2 concentration. So in these two examples here, the black bar being a high CO2, you can see the exchange efficiency decreases when you increase the CO2 concentration. We know CO2 is increasing in the future. So not only do we have this natural variability, but we've also compounding it with changing climate. So we want to know, how are those mycorrhizal associations in wheat affected by atmospheric CO2? And how does it vary between cultivars? How does the function, and then how does the responsiveness vary between the cultivars? So this is the work of one of my PhD students who just graduated last year, Ashley Elliott. And she was looking at the efficacy of mycorrhizal inoculants. So looking at, do they increase colonization? Do they increase mycorrhizal functioning? And she was looking at it across three different wheat cultivars, so Skyfall, Avalon, and Cadenza. So adding in these figures, she's got two color bars. The black bars are the ones that she hasn't added inoculum to. The white bars she has added inoculum to. So these were grown in in a farm soil. So there was a latent spore bank in there. So everything has got mycorrhizas, but we were just adding an inoculum that a farmer might add to the field to see what it would do to those crops. And what she found is when she added the inoculum, all the cultivars increased colonization. But perhaps more interestingly, what she found was the background colonization in the wild soil, or the farm soil rather. It was very variable between cultivars anyway. Then when she looked at the different structures that were produced, this is the percentage of arbuscules and vesicles in the bottom one, she found there was also cultivar variation in the number of arbuscules that were being produced, but this was all increased again when she added the inoculum. And similarly, similar story with vesicles. She got an increase in vesicle abundance in roots that had been inoculated. So that's cool. So the inoculation worked. It increased the abundance of those structures in the roots. However, she then looked at the function. So what were these structures actually doing? And what she found, so the plus and minus again, this is plus inoculum and minus inoculum. She found, firstly, if you look at the minus ones, that plants were getting phosphorus in exchange for carbon. But this was very variable between cultivars. When she added inoculum, it increased in some of them the amount of phosphorus they were able to get, but it didn't necessarily increase the amount of carbon allocation. So it gave clues that there was some there's some effect of cultivar on fungal colonization and on fungal function, and that colonization is not necessarily related to function. So she also found that there was some interest, the carbon stuff was very interesting, that there was no real change in the amount of carbon that was going to mycorrhizal fungi. And this could be down to a number of reasons. I mean, due to the nature of the experiments and them being destructive, it could just be that we labeled at a bad time. Maybe the plant wasn't allocating much carbon to its fungal partner at the point in time we measured it. It could be actually that we just chose a bad fungus to introduce, and maybe we chose one that wasn't perfectly suited to those particular cultivars of wheat. But it emphasizes the issues that are out there and some of the complexities that need to be taken into account when thinking about manipulating soil fungi for use in agriculture. So Tom Thurkel, he took this a step further. So Tom is a postdoc in my lab, and he took this a step further, and he actually did the carbon for nutrient exchange experiments and changed the CO2 concentration. So using the same 3 cultivars of wheat, this is total phosphorus in the wheat tissues. First, you can see it varies between cultivars. And secondly, you can see the CO2 response varies between the cultivars of wheat. Now, this doesn't tell us anything about how mycorrhizas are functioning though. So what proportion of that is mycorrhizal and how is that affected by CO2? So we did the tracing experiments, And basically what we found was these huge differences between cultivars in the mycorrhizal contribution to wheat phosphorus assimilation. There was no significant effect within cultivars. So this seemed to be really a cultivar-driven effect rather than it being a CO2 response. But interestingly, we do have like this slight decrease in two of the cultivars and an increase in the other. So there's a lot of variability there. When you look at nitrogen concentration, this was also affected by CO2 and cultivar. So it appears to be under the higher CO2 concentration tested, we have this nitrogen dilution effect where you get a decrease in the concentration of nitrogen per unit of biomass. However, when you look at the mycorrhizal contribution to that, so it's the 15N label in this figure, you lose that dilution effect. So it seems that mycorrhizas might act to relieve some of that nitrogen, the nutrient dilution effect of the increased biomass that you see at high CO2 concentrations. And we've also seen this in barley. And so there's a couple of papers here that Tom's published very recently that detail some of that stuff. If you look at the carbon, not very exciting, I'm afraid. That doesn't seem to be affected by cultivar. It doesn't seem to be affected by CO2. So the take home really from this is that the carbon in wheat, at least, doesn't seem to be related to the phosphorus or nitrogen assimilation via fungal partners. And so, yeah, there's huge potential for breeding better functionality of mycorrhizal fungi into modern day crop cultivars. So just to summarize then, there seems to be cultivar specific receptivity to colonization in wheat, and this increases with inoculation. Mycorrhizal fungi seem to contribute to wheat nitrogen and phosphorus uptake, and this varies according to cultivar. And this, the nitrogen and phosphorus uptake, it doesn't seem to be linked to the amount of carbon the plant is putting into the fungal network. And that has lots of interesting ramifications in terms of symbiotic function and regulation. But really, I think that the take-home message from my experiments seemed to be that symbiotic function in wheat doesn't really seem to be affected by changing CO2 concentrations, and the variability and the potential that there is in the existing germplasm for functional mycorrhizas in crops seems to be good news for future use of AMF in agriculture. There seems to be huge potential there to exploit these soil fungi for our gain. And I think that's about it from me. So thanks everybody in my lab, collaborators, postdocs, PhDs and technicians, past and present at both University of Leeds and Sheffield, our funding people, and also our industrial partners. And I'm happy to take any questions. I think we've got some time.